All right, so first of all, this is a picture of mitosis. Mitosis is just where we take a mother cell, or just the original cell, and it is going to divide itself into two daughter cells. Now, a lot of misnomers are that the mother cell actually stays in existence and has babies, which is not true. So what is happening is the mother cell literally is dividing its DNA exactly, because you know from talking about DNA replication that we create exact copies of the chromosomes, and then we are going to divide them into two separate cells. And the cytoplasm is divided in half just by pinching it in two. So it is not exactly alike the other cell. In other words, there may be a few more mitochondria in one versus the other, few less endoplasmic reticulum membranes versus in one versus the other, etc. All right. So anyway, so that the cytoplasm is not the same exactly, but the DNA is the same exactly. Now, mitosis in a eukaryote cell is pretty much how we described it with the DNA replication. So it's just the mini chromosomes divided into two. If you're a prokaryote, you have that one circular strand of DNA. So that one is also going to be replicated. It's a slightly different process. It's shown in your book, so make sure you kind of look that over. But it's a circular piece of DNA, and that piece of DNA will be divided into two pieces of circular DNA, and those will be separated into two daughter cells. So really it's pretty much the same, except for the number of chromosomes and some of the physical things that happen within the cell. All right, so the next thing is the purposes of mitosis. So the purposes are mainly in a multicellular organism, that is growth. So you start off as a single cell that was created by your mom and dad's egg and sperm, and that is called a zygote. And that zygote grows into you, the organism that you are, through this cellular process of mitosis. The other thing is repair or replacement of cells that either die or get damaged or something like that. And then the last thing is asexual reproduction. This is of a single-celled organism, like a bacteria, or one of the things that, single-celled organisms that we call a protease, that they like the paramecium that we saw in one of the first labs. Also, this type of asexual reproduction is like a propagation. It's called budding, or sometimes taking cuttings of plants and so forth. Those are a form of asexual reproduction, and that's included in this. Okay, another thing to talk about is something that we call the cell cycle. So if we're going through any process of cell division, including mitosis, but also meiosis, the cell cycle is kind of like a pie diagram of the percent of time spent at different parts of the cell cycle. So in the cell cycle, we have gap 1, synthesis, and gap 2, all of which are part of what we call interphase, and then we have the actual cell division part. So this is just kind of showing you the time spent. Notice that the actual cell division is a pretty short amount of time, even if it was meiosis, which is generally speaking a lot longer than mitosis. There's my cat, sorry. All right, so in gap 1, we'll talk about what happens in each of those areas. And but I wanted to say one more thing about this is the amount of time that it takes for a single cell to do this is anywhere on the order from a couple of hours to almost like 48 hours, something like that. But most of the time it's between 6-8 hours or so that a single cell will actually do mitosis. Notice time 0 up there. Time 0 is when cells are just sitting there. They're not really doing division. They're just functioning. For example, a skin cell is not getting ready to do cell division. It's just acting as a skin cell. And then as the one next to it gets damaged, or it gets damaged, it's going to go through the mitosis process and create two cells to fill the gap of the damaged cell or to replace the cell that is damaged. Okay, so anyway, so cells aren't always going through mitosis, although in some references they say that time zero is really not in existence, that all cells exist in gap one. So if they did that, they'd mostly be just existing at the first part of gap one. So as we go through the process, just kind of keep that in mind. All right.